Hi, this is Rachel and Jesse. And we are going to be talking about the evolution of hearing in amniotes today.
Amniotes are reptiles, birds, and mammals who are classified by the fetuses being surrounded by amniotic fluid in its egg or uterus. 300 million years ago, amniotes vertebrates diverged into leposaurs, which are on the bottom left, and consisted of lizards and snakes, and archosaurs, which are on the right, and consist of crocodilians and birds, and mammals in the top left, which consist of egg-laying, poached, and placental mammals. The ancestral amniotes had a small hearing organ called the basilar papilla. It evolved from the hair cells of vestibular organs. Initially, the basilar membrane only responded to low-frequency sounds. Their low-frequency vestibular hairs became specialized to transduce higher frequencies. This occurred because their ion channels evolved to be electrically toned to specialized frequency. Over time, land vertebrates lost their heavy otolithic membrane, which was responsible for low responses. This resulted in a greater hearing sensitivity.
Alright, so evolution of the middle ear. So this occurred about 250 to 200 million years ago, and the interesting thing is that it occurred independently in all three lineages within about 20 to 30 million years of each other, and each lineage evolved a tympanic middle ear, which is an assemblage of tiny bones that connects the eardrum to the oval window. So in archosaurs and leposaurs, the middle ear bone derived from a bone that connected the brain case to the outer skull. Um, and this extension connects the eardrum to the oval window, and it forms a lever that magnifies sound entering the middle ear. So the stapes right here. And in mammals, it also kind of formed a lever. However, the bones derived from the jaw bones. So as you can see, the reptile has a larger number of jaw bones than the mammals do. And, like the squamosal, or no, the mammal has a squamosal, but the quadrate and the articular became the malleus and the incus. And mammals also have a stape. So they have three ossicles instead of just one.
Of the three lineages, the leposaurs had the least developed ear, and the archosaurs had the most. It was hypothesized that hearing was not a strong selection pressure for the leposaurs because they lost their eardrum. Lizards' hair cells evolved into two different types. One is able to respond to low frequencies and might correspond with their ancestors' hair cells. The other hair cells have more specialized structures in regard to their size and height of their stereovilli and bundles for higher frequencies. Tall bundles with less stereovilli respond to lower frequencies, while short bundles with more stereovilli respond to high frequencies. In regard to the heavily evolved archosaurs, they develop tall and short hair cells. The hair cells rest on the inner sides and the area most distant from the middle ear. The short hair cells lack a connection to the brain and most likely amplify the signal received by the inner ear. Despite all these changes, they on average can only hear from 5 to 8 kilohertz. In regard to mammals, their papilla grew and resembled those of the earliest mammals. Therian mammals evolved cochlea, and their hearing organ also evolved into a full circle roughly 120 million years ago. Larger mammals also tend to have a lower upper frequency limit than smaller mammals, but most mammals have a higher upper frequency limit than leposaurs and archosaurs. In some cases, like bats and whales, their hearing extends into ultrasonic frequencies because of their stiff connective tissue. However, humans have softer connective tissue, which allows them to hear lower frequencies.
All right, so another really interesting thing that evolved with regards to hearing is the ability for hair cells to produce active movements that further amplify sound stimuli. So this is called active bundle mechanism, and it helped our ancestors overcome surrounding fluids that resisted movement. So what happens is sound stimuli enters the ear and causes potassium ions and calcium ions to be released, and the calcium ions open these transduction channels that increase the speed of the channel closing. Um, so this increases the distance of the hair cells that the hair cells move, which increases the sensitivity of it, which amplifies the sound. Um, Another interesting thing that hair cells do is that outer hair cells can change their length if exposed to alternating electrical fields like those that occur when the hair cells respond to sound. And this force causes the organ of cortices to move up and down, um, which allows quieter noises to be detected, even as far as ultrasonic frequencies in bats and whales. So this is due to changes in a protein called prestin, changes in the molecular configuration, and this evolved independently in all three lineages um, to form specific configurations, which allowed either more sensitivity to hearing or less, depending on the species. But all were due to the changes in prestin. Um, a final thing is that in birds, the movement of short hair bundles also can lead to the amplification of sound. So that was something cool that evolved. And yeah, so the thing about this amplification is that it can lead to spontaneous movements of the hair cells, which are called spontaneous otaconic emissions. And this enables remote sensing of what is going on in the inner ear, so that's pretty cool. Um, and yeah, I think that is about it. Thank you for listening. We hope that you learned something new about the evolution of the ear in amniotes. Yeah.